Hello everyone, I'm here by the names of Sessione, and I am here to present about the assignment that you have been given, the module which is immunology and nutrition aspect of disease. I was expected to start from 9 question and then to 14, so I am from human nutrition and dietetics, level 3, so let's continue with the presentation. This number 8 question, it was asking how does the complement system support immune function? Firstly, complement system is the system which is among immune system, which is composed of many plasma proteins which react to one another. This is to mean they make whole in pathogen, and then the protein these antibodies they get inside the pathogen, which at the end which will make pathogen burst. Uh, After bursting, already pathogen is dead or it is killed, and also they induces the series of inflammatory response which is there to help fight infection. This is to mean by inducing series of inflammatory response, this is just calling upon the immune cells to fight against that pathogen and to prevent further inflammation. So this is the way they do it. They do it by firstly they opsonize pathogen. Opsonizing pathogen to just making more attractive to phagocyte, to just representing this pathogen to the other immune system, like immune cells, like phagocytes, to the time that they come. Also, there is lysis. I have said lysis is where the the antibodies come into the pathogen, and then they make it burst, which is what you call lysis. It's already what you call lysis of the bacteria. Also inducing inflammation, just inducing inflammation, just like calling upon the other immune cells to the site of infection, just for fighting and for prevention of furthermore damaging of the tissue. Here is the mechanism by which complement proteins work within the system of immunity. Here, the first step is the activated complement proteins, which forms complex of the protein. This complex of proteins, they create holes in bacteria or cell wall. Yeah, yeah, it is understandable because, uh, yes, second one, water and salt, they diffuse in the cell wall, which is in the whole of the cell, which is made by that complex proteins. Yes. Then the third one is that bacterium swell and eventually burst. After inversion of the water and salt within this bacteria, bacteria will swell, it will become bigger. After becoming bigger, it tends to be burst. After bursting, uh, it will lead to the rise of bacteria. Okay, here we are having one question which was asking what are the natural killer cells and how do they combat infections? Firstly, natural killer cells, they are a type of white blood cells that can firstly recognize and secondly that can kill infected cells. So as you know, in the immune system, there is firstly recognizing and also the engulfing or killing the infected ones. They do this by firstly to recognize the protein surface of infected cells. Secondly, they come to produce cytokines which help to activate other immune cells. So, 10th question. which was asking, how do regulatory T cells maintain immune tolerance? These regulatory T cells, they are immune cells, or they are white blood cells that prevent immune system from attacking their own tissues. Uh, they have to suppress the activity of other immune cells. It means they control the activities of the other uh, immune cells. Number 12 question was asking the role of dendritic cells in immune surveillance. These dendritic cells, they are specialized immune cells that can engulfy the first one and they process antigens. They then present these antigens to the cells, which can help to activate the immune response. This is to mean that first dendritic cells they are able to engulfy, which is to mean they put in cross surface pathogen which is embedded, and then they secrete some enzymes which digest that pathogens, and also they are there to process antigens, which is to present the antigen to another cell or for activation of immune response. Uh, the certain question it was asking the way the vaccination stimulate immune system. Firstly, vaccination is the introduction or provision of weakened or inactive form of pathogen in the body. This involvement or invasion of inactive pathogen in the body, they stimulate immune system to produce antibodies and T cells that can protect against future infection with the same pathogen. This is to mean after this vaccination, there's production of memory cells which will be able to counter which will recognize the second invasion of this pathogen so that it would not cause a disease. The last question it was asking, it was 14, which was asking what is the important or what is the immunological memory and why is it important? As you have said in previous questions, number 13, it was asking the ways through which vaccination work in immune system. This immunological memory, it is ability of immune system to remember past infection. This is to mean if the infection comes faster and then later at the second encounter, it will be recognized by these memory cells which have been produced so as to prevent the damaging or inflammation or the disease causing. Uh, this memory cells allow immune system to respond more quickly. This is the first one, and effectively to the future infection with the same pathogen. And also, the why this is important is that it is the basis of vaccination, as I have said. So thank you. This is my presentation. Thank you for your attention. God bless you.